Assalamualaikum and have a good day everyone. So in this video we will learn chapter 5 response in plants in subtopic 5.2 and 5.3. Okay, 5.2 is phytohormone and 5.3 is application of phytohormone in agriculture. Okay, let's discuss more about the phytohormone. What is phytohormone? Okay, phytohormone or plant hormone are chemical substances that stimulate and coordinate responses in plant at low concentration. Generally, phytohormone are synthesized in a certain plant organ and transported to a target organ through the flower. Examples of phytohormones are auxin, Gibraline, Cytokinin, FCCC and ethylene. So this is the overall picture of hormonal interaction influence plants growth and development. You can see the cytokinin, the Gibraline acid, okay, to control the cell division, FCC acid to control the stomata, ethylene and auxin, development of adhesion zone, auxin and Gibraline to control of stem elongation, cytokinin, growth of young fruit, and ethylene, ripening of fruit. And last one is auxin, Phototropism of fruits. So let's discuss more detail about every part or every phytohormone. Auxin, functional in phototropism and geotropism, stimulate development of apical dominance in shoot and roots, stimulate growth and elongation in roots and shoot cell, stimulates development of adventitious root at the stem of stem cuttings, stimulates cell division in cambium rings during secondary growth, inhibit abscission of fruit and young leaf. Abscission means pengguguran daun dan buah. And lastly, inhibit growth of lateral buds. So this is the picture of effect auxin to elongate the structure of shoot. Okay, after this we discuss detail more about the auxin. Gibraline stimulate growth and elongation in the stem cell, stimulate development of leaf, fruit and flower, stimulate development and germination of seeds, causes growth of flower in dwarf plants, and lastly, inhibit development of fruits. So, Gibraline effect in the growth, elongation and development of seed. Cytokinin stimulate division and elongation of root and stem cell when auxin is present, stimulate seed germination, inhibit development of apical dominance, delay leaf senescence, stimulate growth of lateral buds. So this is the relationship between the cytokinin and auxin. Auxin oppose the development of axillary buds. Okay, so the movement of auxin is from the shoot to the roots, but the cytokinin oppose. Okay, transport outwards. The key concept for cytokinin is cytokinin will increase more mitosis. So more mitosis will increase more cell. So that will, if there is more cell, it will stimulate the plant growth, because the cytokinin stimulate the cell division. FCCC inhibit growth of plant, stimulate abscission of matured fruit, leaf and flower, induce seed dormancy, induce stomatocrosis during drought season, and inhibit growth of buds and seed germination. So FCCC is used for the plant to abscission. Abscission means pengguguran daun. So induce the seed dormancy, induce stomata opening, especially during the drought season. Yes. Okay, ethylene. Ethylene stimulate ripening in fruit, stimulate senescence process in plants. Senescence means pengguguran daun. Abscission of leaf. Okay, senescence means the last stage of the fruit, the plants. Okay, such as abscission leaf and fruit. Okay, this is how the ethylene simulate ripening of fruit. Okay, for the fruit, ripened fruit, okay, there will the fruit are acidic or sour, have a lot of starch, have chlorophyll, more chlorophyll. That's why it's in green in color. There's a lot of pectin and large molecule inside the ripened fruit. Okay, and then the ethylene will stimulate the enzyme. Okay, from acidic or sour fruit. Okay, the enzyme kinase will convert the sour into the neutral, the acidic into the neutral. The amylase enzyme will convert hydrolyzed starch into sugar, make the fruit become more sweet. Hydrolase enzyme will hydrolyze chlorophyll, more chlorophyll to less chlorophyll. The pectinase enzyme will convert pectin, they make the plant hard, to less pectin, make the ripened fruit more softer. And hydrolase enzyme make hydrolyze the large molecule into the small molecule and increase the fragrance of ripened fruit. So this is the overall function of the phytohormone, such as the process of germination. Okay, only the Gibraline involved in germination. Growth and maturity, Gibraline, auxin and cytokinin. During flowering, Gibraline, auxin, cytokinin and ethylene. Fruit development also involve Gibraline, auxin, cytokinin and ethylene. But the efficient ethylene and FCCC. Okay, and last one is seed dormancy, is FCCC. So this is the life cycle of the plant, from germinate until seed dormancy. Okay, after germinate, we grow to the maturity, and then the plant will flowering, and the plant will form the fruit, and abscission, and last one seed dormancy. Okay, let's discuss about the effect of auxin on the growth response in detail. So the plant respond to the stimulus in the form of growth. Okay, so we have learned about the response. Okay, in 5.2 and 5.1. Okay, the plant respond to the stimulus in the form of growth, tropism. So the growth response is controlled by the auxin. Okay, this is the experiment of coleoptile to show the effect of auxin on the growth response. This is coleoptile. Okay, coleoptile. The structure of coleoptile at the seed. Okay, so in this experiment 1, okay, the tip of the coleoptile remove. Okay, above the elongation zone, so the tip of coleoptile remove. Okay, this one, experiment 2, the tip of coleoptile is placed, and the tip of coleoptile is placed on the agar block. This is agar block. And the experiment 3, the tip of coleoptile is placed on the top of agar block. Okay, but the experiment 3 is different with the experiment 2. Okay, let's discuss more of the result of the experiment. Okay, So what happened after in the experiment 1? What happened after 2 hours? Okay, look at to the picture. No growth is observed after 2 hours because there is no tips of the colloidal. So this is show that no auxin produced because the tip was removed. But when the tip is put back on the colloidal, the colloidal will grow outwards. Okay, we put back the tip of the colloidal, and we can see that the colloidal will grow outwards. Okay, let's see the result of experiment 2. Okay, after 2 hours, we put the agar block. Okay, we put this agar block into the colloidal. Okay, the agar block is placed off center. Okay, center of the colloidal. What happened? The colloidal grows outwards. That means there are an auxin inside the agar block. Okay, so auxin inside the agar block because the auxin can diffuse across the agar block. So this agar contain auxin. So auxin will diffuse into the colloidal. And the second one, the third one experiment. Okay, the agar is put half of the colloidal. Okay, we put half so that the auxin will receive by the half of the colloidal. So it will make the growth of colloidal is curved because only this side receive the auxin. Okay. So inference that can be made based on the observation from the experiment. So the experiment showed that auxin which has been synthesized at the colloidal tips, okay, stimulate elongation. Auxin distribute influence the direction of shoot growth. Okay, uniform distribution of auxin cause the colloidal to grow outwards. Okay, auxin will influence the direction. Uniform distribution of auxin cause the colloidal to grow upwards. The area that receive more auxin will grow faster, cause the colloidal to curve to the side of receives less or none auxin. Okay, so this is the auxin. Auxin produced at the tips of the colloidal. Okay, and then we diffuse to elongation zone. Okay. So it will make the growth of plant grow longer. Okay, how about the agar block that place only half of the colloidal? Okay, the auxin produced at the tips of the shoot, and then it will diffuse to the cell elongation. Okay, this is the uniform distribution of auxin. But if we put only half side of the colloidal, the auxin will accumulate at the one side of the colloidal. Okay, when we put the colloidal place on one side, so the side that receive more auxin here can elongate more. That's why the colloidal growth is curved. Okay, inference that can be made based on the observation from the experiment is tropism is closely related to auxin distribution. External stimuli such as light and gravity influences the auxin distribution in plant. Auxin produce different effect of the cell in the shoot and the cell in the root. High auxin concentration stimulate cell elongation in the shoot, but also inhibit cell elongation in the root. Okay, this is the graph the graph to show the effect of auxin towards elevation of shoot and root. Okay, so this graph of shoot, high concentration, this is high concentration of auxin will stimulate. Okay, high concentration of auxin will stimulate the cell, but okay, in the roots, okay, inhibit the cell elevation. Okay, so the concentration of auxin will inhibit the roots. Okay, but high concentration of auxin in the shoot will stimulate. That's why the different pattern of the graph. Okay, let's discuss more about the role of auxin in the phototropism. So the response direction of shoot tip depends on the direction of light stimulus. The distribution of auxin in the shoot is uniform. If the shoot responds to the light from all directions, it's called the shoot to go upwards. So when we put the colloidal, okay, in the uniform distribution of light, okay, cahaya datang daripada seluruh arah, and the shoot will grow straight. Okay, the cell both sides of the stem elongate equally. But if the shoot are exposed to the light from only one direction, such as we put the colloidal in this side, the auxin will move away from the light. Okay, the auxin concentration is higher on the shaded side. The concentration of auxin become uneven on the shoot. The cell in the shaded side elongate more than the cell in the bright side. As a result, the shoot bend toward the light. Get to the picture. So we put only half side of the light stimulus. Okay, the light from this part. So the auxin will move to the shaded. Okay, move to the shaded region. Okay, so more concentration of auxin in the shaded region. So it will make the more elongation of the shaded region. That's why the shoot bend toward the light. Okay. So that's why the shoot show positive phototropism. So this is the several experiments to show the effect of the auxin on phototropism. What happened after 2 or 3 days? So this experiment is controlled. This experiment, the collector is cut. Okay, and then this experiment is the collector is covered with the opaque cover, and this collector will cover with this transparent cover. And then this experiment, the base of the collector will cover with the opaque object, and then the collector is separated by a piece of agar block, and the experiment is separated by beaker. So after 2 or 3 days, okay, for the control experiment, of course the collector bend toward the light because that is control experiment. For number 2, the collector is cut, so no growth because no collector, no auxin. Okay. For the third experiment, the collector grows outwards because the opaque cover, okay, will cover the collector from receive from the sunlight. So opaque, okay, object legap ni we prevent the sunlight. So the collector will grow outwards. Okay, the auxin will produce inside the collector and diffuse to the elongation zone. But when the experiment is treated by the transparent cover, the collector will receive light. So the auxin will move to the shaded region, the other side. So it will make the collector show the phototropism, positive phototropism. So for the cover with the opaque object is base. Okay, it does not affect the collector auxin distribution, but for the agar block agar block is the medium that allow the diffusion of auxin. So auxin we produce here and we diffuse across the agar block. But mica is the substance that prevent the diffusion of agar. So no growth is observed. So the role of auxin in geotropism. Auxin also control plant response to gravity. Seed germinate horizontally in the soil due to the pull of gravity. Auxin pulls at the bottom side of the tips and the roots, and the roots grow downwards and following the pull of gravity and exhibiting positive geotropism. Meanwhile, the shoot grows upwards against the pull of gravity, exhibiting the negative geotropism. Look at to the picture. Okay, so this is the seed germinate horizontally in the soil. This is the seed, germinate horizontally. Okay, and then in shoot, okay, look at to the shoot. Okay, high auxin concentration cell in the at the bottom of the grow faster and allow the bend shoot to bend upwards. Okay, this is the auxin. Okay, auxin we produce on the tip of shoot, and the high concentration auxin will more accumulate at the bottom part of the shoot because of the gravity. Okay, so that's why this will make the shoot will bend upwards because high concentration of auxin will stimulate the shoot. Meanwhile, in roots, the production of auxin inside at the tip of the root and accumulate at the bottom part because the gravity, but the high concentration of the bottom root inhibits. Okay, it's differ to the shoot. Okay, shoot will grow faster, but in the root inhibits cell ligation. So that's why the high concentration of auxin at the root will inhibit. So that's why the root bend downwards. Okay, the root bend downward to show the positive geotropism, while the shoot will show the negative geotropism. Okay, this is the application of phytohormone in the agriculture. How was the seedless fruit produced? Okay, right. The watermelon is there is no seed. There is no seedless fruit. So the application of phytohormone in agriculture to produce seedless fruit is called parthenogamy. So phytohormones are important chemical that are directly involved in the development of fruits, flowers, stems and roots. Phytohormone can exist naturally or be synthesized in the laboratory. There are phytohormones that can be extracted for multiple purpose in the agriculture. Okay, this is also the application of phytohormone. Okay, the ethylene gas. So let's discuss more about the auxin. So auxin encourage growth in crop production, roots growth and woody plant stem for the horticulture crops, or produce fruit without seed via the parthenogamy. So this is how the seedless fruit produced, and through the parthenogamy we use the auxin spray to the unfertilized flower, and the flower will develop into the seedless fruit. Used as a weed killer. Induce dormancy in potatoes during storage or distribution. Or promote growth of low-lying and large plant. Next, Gibraline. So the external application of Gibraline can also enlarge fruit size in grapes. Okay, and we can see the structure of grapes. Okay, after treat with the Gibraline, we produce larger size. Treat mutated dwarf plants to grow to normal height. Promote rapid elongation of flower stem. Used to produce larger grapes. Promote germination of seed such as salad, oats and tobacco during low temperature and low light intensity. cytokinin. So cytokinin we use in tissue culture technique, promote cell division and differentiation of cell. Tissue culture technique used together with auxin to form plant organs such as root and stem, used to delay the leaf senescence in the newly cut flower. So cytokinin is used in with the auxin in tissue culture technique. We have learned the tissue culture technique during Form 4. Ethylene. Ethylene used commercially to promote maturation of fruit quickly and evenly, promotes the simultaneous flowers in plant in the field. So this is the effect of ethylene, okay, naturally produced by the during the ripening of fruit. Okay, so ethylene gas we use to stimulate the ripening of fruit. Okay, the ethylene will evaporate in the form of ethylene gas. And last one is FCCC, will inhibit germination and growth. Okay, thank you.